Okay, so due to popular demand, I am making another video. This video is for note number 10, which is the fluid mosaic model of the cell membrane. Okay, so what you boys need to know is you do need to draw a diagram similar to what I have on the screen right now, similar to what I had on the whiteboard in class. You do need to be able to draw one of these diagrams for the exam, and you also need to be able to label each part of it. So I currently have labeled the extracellular fluid, which is the outside of the cell, and then you have the cytoplasm here, which is the inside of the cell. We also have a glycolipid. And glyco references a carbohydrate. The lipid tells you where it's attached to. So in this case, this carbohydrate is known as a glycoprotein. And that's because that carbohydrate is attached to a protein, whereas these pink ones here are attached to a lipid. So they are also known as glycolipids. You also have different proteins that are within the cell membrane. There's two different types, and the main difference is how far it goes through the membrane. So the peripheral protein here, this one, it does not go through the entire membrane. You can see it either, this one in this case just goes through the side facing the cytoplasm, but there might be some proteins that are only facing the extracellular fluid. That would still be a peripheral protein. But proteins that go through the entire cell membrane, like this one right here, are known as integral proteins. Now, you'll sometimes read it, and some books will call them the transmembrane protein. That is the same thing. So the cell membrane is also known as a phospholipid bilayer. That's because it's made out of two phospholipids. So one phospholipid looks like this, and there's two. One at the top, one at the bottom. So the two of these together make the phospholipid bilayer. So you also have a phospholipid. Each phospholipid is made out of two parts. It's got a, if I can fit it, it's got a hydrophilic head. Hydrophilic is also another way to say water-loving, and it's got two hydrophobic tails. So we've got two hydrophobic tails. If you haven't already, I would take the time to actually draw this out because I'll actually reference it more than once in the note. So it's definitely worth your time to draw out a membrane so you can draw out the different processes. So if you turn to note 10, which is the fluid mosaic model of the cell membrane, okay, this is our note. If we scroll through, I've got a bit of a cartoon diagram here. And our cell membrane is known as a fluid mosaic model. Okay, so there's a different reason. There's two reasons why it's called fluid and mosaic. Starting with fluids, we have to think about the hydrophobic tails. These tails right here, those hydrophobic interactions are what help the membrane stay together. So if we remember back to our last unit, there's two types of lipids. We've got saturated and unsaturated fatty acids. If our lipids were all saturated, there would be no kinks. Without any kinks, these phospholipids would be able to stack very tight against one another, leading to a very rigid cell membrane. However, that doesn't actually happen. Normally, your fatty acids have many kinks, so they're made out of unsaturated fats. Sometimes some membranes can have saturated fats, so I'm not saying they're always unsaturated, but having that unsaturated fat allows for the fluidity in the membrane because they don't stack as tightly. And because they don't stack as tightly, it allows for smaller molecules like water to go across the membrane. So, as you all know, the cell membrane is important as it acts as a barrier between the environment of the inside and the outside of the cell. The first 2 points are generally review, but these points that I've highlighted here, this is the new part that you likely haven't talked about yet. Why is it called fluid? It has to do with the flexibility, the ability for our membrane to change shape based on saturated, unsaturated fats, things like that. And the mosaic comes from the idea that it's made out of so many different parts that come together. Now, and not every section has to have five peripheral proteins or 4 integral proteins. It's actually very random. It depends on the cell function, what its needs are, things like that. So the mosaic is just the idea that it's a combination of all these different parts. Now our membrane's made out of the phospholipid, which I've showed you previously in my cartoon drawing, but this breaks it down from the chemical formula perspective. What you need to know is where is the hydrophilic head, where is the hydrophobic tail, and what makes up these components. So in this case, you can see you've got phosphate and glycerol. That's what makes up the hydrophilic head. And then the hydrophobic tail, it's made out of 2 fatty acids. They don't always have to be saturated, one unsaturated. That's just the example here. But what you need to know is the hydrophobic tails are 2 fatty acids. That's also why it's not water loving, because fats and water don't really get along. Then we have our membrane proteins. So a lot of this stuff, you just need to know these 5 processes. You won't need to know specific examples. You will have to do specific examples next year. So the function of a membrane protein: 1, transportation. Integral proteins, they face both the outside and the inside of the cell, which are perfect passageways for things to go through. As well, proteins are enzymes, so they help carry out many reactions. And next year during biochemistry, you will learn so many of these reactions, and it's super cool. They are also receptor sites. That's important because these can be chemical messengers. They help identify different things, help cells communicate to each other, which leads to my next point, which is cell adhesion. Sometimes if cells need to communicate with each other or come close together, the proteins will facilitate that. There's also the attachment to the cytoskeleton. For those of you who remember mitosis, we talked a bit about the cytoskeleton, but this helps maintain cell shape. So if I scrolled up to the picture at the top, you'll see the cytoskeleton, these small little yellow components at the bottom right here. Right, so the proteins help attach to it, and it helps hold the shape of the cell. That is the attachment to the cytoskeleton. That would be point number 5. And there's 2 types of proteins. You have your integral proteins. These insert into the membrane, also meaning that they face both the extracellular fluid and the cytoplasm. And then you have peripheral proteins, which are just to the surface of the cell membrane. So they can either face the ECF only, or they can face the inside, the cytoplasm, only. And then you have carbohydrates. These are named based on where they attach to. So if they are attached to a protein, they are a glycoprotein. If they're attached to a lipid, they are a glycolipid. The glyco refers to the presence of a carbohydrate. So you'll hear, like glycogen, you'll hear words like glycogen. Glycogen is a storage molecule of carbohydrates. Glyco, again. And the main function of the glycoprotein and glycolipids is the cell-cell recognition. So this is another way that cells can communicate with each other. You can now start to see how so many of these chemical interactions are so important for the overall biological functioning of any system. And then I threw the picture back in for you to help picture it all after going through the knowledge there. Okay, so the fluid quality of membranes. Membranes are never static. Nothing in a biological system is ever static. Everything is always dynamic. Things are always happening. The hydrophobic interactions keep everything together. And sometimes things can move laterally in a membrane. The membrane is in a plane sort of fashion, so things can move around. Yes, it surrounds the entire cell, but things can move, or proteins can cause different reactions for proteins to shift. There are many protein pathways, which you will learn a ton of as we go forward if you continue this field. Temperature also affects the membrane. So if it's really cold, it's going to change permeability. Permeability, if you remember from the lessons with diffusion, is the ability for things to move through. So what can and can't move through will really change with temperature. The enzyme proteins will also differ because proteins like to function at an optimal temperature. And if it's not there, they're not going to function, which means those enzymes will not catalyze the reactions we would like. There is cholesterol. We're not going to worry too much about it, but for your own knowledge and for fun, cholesterol helps with fluidity. So cholesterol you saw in the diagram above, I'm not going to worry about having you boys draw it, but cholesterol helps with fluidity, and it either helps keeping them from packing too close, but also from going too far apart. So it helps with the overall fluidity. Okay, so now we're going to talk about transport across the cell membrane. I think I'm going to make a flow chart for this because it'll help go through it'll help organize your information. So let's call this transport across the cell membrane. Okay, and there are two types of transport. The first one in your note is passive transport. And this is something you already know of very well. This is driven by diffusion. So if you remember diffusion, if you don't, I'm going to tell you about it right now. So don't you worry. I'm a little far in this note. Okay, diffusion, it's the movement of molecules from a high concentration to a region of low concentration. This drives passive transport. Passive transport happens very naturally. It does not need energy. It's like its name, it's passive. Okay? The osmosis is the diffusion of water across a semi-permeable membrane. This part here, the solutions, you will talk about this in grade 12, but it's also good to know how cells react to different solutions, because depending if there's more salt in a solution or less salt, the water will move in a certain direction. I'll leave that for you to go through in your own reading, so I tell you what the concentrations are and which way the water will move. I'll leave that to you for your own leisure because that's more for interest. Okay, so let's go back to passive transport. So I'm going to move to my transport here. This is driven by diffusion. It's no energy needed. Again, no energy needed because it's very natural for it to happen, right? And a type of passive diffusion that we talked about, or I sort of mentioned, is osmosis. And that is the movement of water from a high concentration to a low. And these square brackets are a short form for concentration. You'll likely see that in chemistry as well. Okay, so going back to our note, you have another type. So that normal sort of passive transport that occurs, you have diffusion first of all, okay? And then you have something called facilitated diffusion. Facilitated diffusion is facilitated by two different things. You have either a protein channel or a carrier protein, and they work in two different ways. So if I go back to my diagram here, a carrier protein works sort of like an opening and closing mechanism. I didn't draw out the phospholipid bilayer here, but imagine this is an integral protein. Okay, and these triangles are moving from a high concentration into a low concentration into the cytoplasm here. So there aren't many triangles here. There's like one versus the three here. So the triangles are moving from the outside to the in. So the protein will open facing the ECF, and then it'll close and open again facing the cytoplasm. So here we have the little triangles moving from a high concentration to a low concentration through an integral protein. And this is known as carrier proteins. And this is a part of facilitated transport. So putting that here, you've got passive driven by diffusion, and you've got facilitated